This part of the assay will be performed under sterile conditions, so the next steps will be done in a cell culture hood. First, gather all the materials you will need to prepare your plates. Start by removing the well template from the kit box and setting that aside. The other components of the assay you will need are a well stand, which is optional, microcentrifuge tubes, cell culture plates, microcentrifuge tube rack, appropriate pipettes, cell media suitable for your cells of interest, well reservoirs, a pipette aid, a vortex, and the cell suspension solution. The first thing to do is to prepare your cell culture plates for adding material. Remove them from their sterile containers and number them accordingly. It is recommended you number both the lid and its base in case they get separated during the assay. The second step is to pour 10 to 15 ml of cell media into a well reservoir. Then, using your multi-channel pipette, transfer 100 microliters into the exterior wells of each of your plates. The 36 wells that make up the outer ring of the plate are unmarked on the well template in white. The purpose of adding only media to the outer ring of wells is to avoid evaporation of the sample wells during the course of the assay. The next step is to vortex your cell suspension solution briefly and pour the contents into a fresh well reservoir. Then, using your multi-channel pipette, attach 6 tips and transfer 90 microliters of the cell suspension into each of the interior 60 wells of each of your plates. The last step is to place the plates under the appropriate growth conditions for your cells and allow to acclimate overnight.